I worked with Felicia in the Tang lab, and my project used GFP as a reporter system to look at the levels of fluorescence and see if they changed when I added a small piece of RNA to a couple of target genes to see whether there was an interaction.
I was working under Sarah in the Tang lab, and the title of my project was the creation and evaluation of second-generation meningococcal vaccines. I picked the project after going to bacteriology lectures and the practicals and getting a feel for what kind of things you would do in a bacteriology lab.
Yeah, similarly, I really enjoyed the lectures that we had on bacteriology in second year, and then the practicals were really fun with the different types of bacteria on the agar plates, and there was a GFP practical, which I found really interesting, so I really wanted to look at something in that area.
Well, it definitely helps in terms of understanding the different techniques, because you read about them and you know what they're to do with in theory, but you don't really understand the practicalities until you actually do one for yourself. And with that comes a greater understanding of in what situation such a technique would be helpful, and what kind of controls you'd be wanting to use. And that comes in handy for reading research papers, which will come in handy this year.
Yeah, I agree. I think as doctors in the future, it's important to be able to understand where things have come from and how they've developed. And if you read papers and things, it's really good to appreciate the science that's gone into it. And doing a research project is a really good way to give you a little bit of grounding in that and to see you've got a bit of first-hand experience in working in the lab. I think it definitely does, because you go from second year where there's a list of everything you need to know to third year where it's up to you and what you find interesting. And I think the lab project's a really good bridge between them. So you start because you know what you have to do for your project, but if there's an area you're quite interested in, you can discuss it with your supervisor and say, oh, can I go along this route instead, or how can we try this one? So it's quite a good way to learn, to start to channel where you want to go with your research and reading and things.
I guess technically towards the beginning, when things didn't work first time, and then that was quite frustrating initially, and having to persevere and try different vectors and different primers and things like that, that was quite challenging in terms of things not working. But then it's also the independence as well, the reading around the subject and the time management.
Yeah, I'd agree with Sasha. I found the cloning and repeating things a couple of times, that was challenging. And also the first lab meeting that we had to do, so when you're in a room with all these scientists and they're all really, really clever and know exactly what they're doing, and then you go up and present, and then I had drawn my plasmids on paint, and they were quite not very well developed, but it was really useful to talk, and then once you get over it, it's fine and it's not nervous, and they'll give you really great feedback and help you and point out things you might not have thought about. It's just a really nice environment to learn from. But it was a bit scary for me before I went in and did it for the first time. So normally there's a list that gets published in Hillary of titles and supervisors, and you'd have a look down the list and email supervisors that you think are interested with projects that you think you'd like to do, and go and visit the labs, and then you would rank the labs in order of preference, and then the labs do the same with students, and then they match you up according to that. But you can also arrange it privately. If there's something you really, really enjoy, you can email a lecturer or a tutor or a supervisor and ask, are they willing to take an FHS student, and then go and visit their lab that way and ask and see and sort it out yourself. So both can be done. I would say it's really important to go and have a look around a lab and get a feel for it and the people there, and meet your supervisor and see if it's somewhere you'd like to work, because it's quite a long time you're there for.
And pick something you're interested in, because 8 weeks is a lot of time to commit to one thing. And it's a lot easier if it's something you enjoy, because you want to read about it and you want to put the effort into your experiments.
Yeah, we went to Stockholm for the IMID conference, which was 5 big labs from all around Europe that work on bacteria mainly. There was some viral talks, and then the interactions between the hosts and the pathogens. So there were people from Stockholm, from Germany, from France. There was a London group. We came from Oxford. It was amazing to see all these different scientists talk about what they were researching.
The talks were really interesting, but also the discussions that would follow the talks and the issues that people would raise there were really interesting as well. Well, ours were really good in the sense that they would talk us through different protocols and make sure we understood it, maybe run through it with us physically the first time, just to give an example before we were left to get on with it ourselves.
Yeah, that was really useful, and to put up with some of the silly questions that I asked. I asked a lot to Felicia, and she was really helpful, and she'd go through them with me and answer them again and again when I'd ask them again and again. But eventually you learn, and like by like the 6th week you can do it all by yourself and it's fine, and there's not as many questions. It's brilliant. I've really enjoyed my time here, and yeah, the history associated with the place is quite amazing, and to think that I've been able to work here for a little bit is really cool.
And there's so much going on within the building, like people are working on such diverse things. It's really interesting going to the seminars here and finding out about it.